Okay, folks, we are going to get started with this breakout session as we continue this fantastic day of science and engineering here at MIT for the HEALS initiative. I am delighted to get to introduce and chair the Biosphere breakout session today. My name's Matt Shoulders. I'm a professor of chemistry, and really happy to see all of you here for this important topic. You made your way all the way over. We really, really appreciate it, and it's a beautiful new space here, so I hope you're enjoying it. Today, I'll start with just a few brief comments of my own and then hand it over to a fantastic cast of 4 speakers whose names you could see on the prior slide, but we'll introduce them along the way to tell you about the topics, to tell you about really amazing research and sort of the future of this field, understanding how living systems power life on Earth, and I'm really excited to hear their talks. When the talks are done, and this is important, when the talks are done, we're planning to have about 15 minutes, 10 to 15 minutes for a panel discussion. That's why we have chairs here. So think about the questions that you want to ask these fantastic faculty, because if you don't, I'll have to ask all the questions, and your questions will be far more interesting. So we'll have time for that afterwards. Okay, so why do we have a Biosphere breakout at the MIT Health and Life Sciences Collaborative? I hope and think that the answer should actually be obvious, right? But the interface between life sciences and health is critically important. But the health of people, the health of the environment, the health of the globe, the health of the planet, life sciences are central to solving the major problems that health in all those spheres depends on. The life sciences are absolutely central to solving the key challenges that we face. So these range, of course, from rapid climate change that we have to adapt to, and we have to figure out how to also halt or slow. Life sciences are these challenges that face the life sciences, include enormous increases in demand for food, for resilient agriculture, and for healthy food, right? We also face the damage done to deteriorating environments and the effects on well-being caused by all of that. And really, the life sciences are key to solving these challenges, whether you talk about synthetic biology, plant science, microbiology, environmental science, ecology. The viable solutions, the globally deployable solutions to these types of challenges, to the health of our world and the health of humans, are going to come in a meaningful way from the life sciences. And we also, along the way, as we come up with these solutions, we have to think about ethical and equitable means of deployment right from the start as we think about inventing these solutions. So MIT is a leader in this space. You've been hearing about lots of amazing biomedical research and tremendous discoveries being made. But at this interface between the life sciences and the biosphere, the life sciences and the climate, life sciences and agriculture, there's also huge opportunities for impact on health. In fact, even sort of small incremental improvements in these spaces can have the sort of massive effect on human health and planetary health that would elude most types of drugs that you could dream of inventing. So we think this is super important, super relevant to the HEALS initiative. And today you're going to hear from these 4 faculty about both challenges and solutions to problems at this interface between life sciences and the environment and sustainability. And you're going to hear them talk about challenges, that are facing our globe and all the way down to individual communities, right? And the talks today are going to start at the molecular scale, as we first start by talking about photosynthesis and the basics of photosynthesis and how it powers the entire planet, how we can maybe understand it better and improve on it to find great solutions. We'll go from the molecular scale to the scale of crops and actually providing food for the world. We'll talk also today about how the changing climate and the interface between the climate and green spaces has huge impacts on human health and human health equity as well. And then finally, at the end, we'll go back to the global scale, and we'll think about how tiny microorganisms play a huge role on Earth. And it's a very fascinating area there as well. You're in for a great treat here with these 4 wonderful faculty, and I should get off the stage so you can hear from them. Our first speaker is Professor Gabriela Schlau-Cohen. She's the Robert T. Haslam and Bradley Dewey Professor of Chemistry.
All right, well, thank you, Matt, and thanks to all of you who made this journey over to enjoyed this view and these talks. So Matt introduced us to photosynthesis. And photosynthesis occurs in a wide variety of organisms and a wide variety of environments to collectively generate energy on an enormous scale. Now, behind this feat is a series of remarkable features. For example, if we take this corn in this crop field here, one question that you may have wondered yourself, and is actually a central question in research in my group, is why is it that plants like this one don't get sunburned? So plants don't have to put on SPF 50 when they go outside. They stay in the sun all day, but yet, except in a few limiting cases, they don't get sunburned. Now, the reason for this is they have their own built-in molecular machinery. It serves the function of sunscreen. In my group, we're interested in understanding the mechanisms behind this molecular machinery, both for fundamental knowledge and because of this plot here. Shown are yields for 4 major cash crops as a function of time. And what we can see over the next decades, with predicted demand in dotted lines and predicted supply in dashed ones, is that there is a growing mismatch. And not having enough food is a huge issue for human health. Turns out we need to eat. And so that means that one of the biggest health challenges facing us is figuring out how to bridge this gap so the supply meets the demand. One way to do that is to increase the supply. Fundamentally, the food supply comes from photosynthesis. But photosynthesis operates at a measly 1 to 3% efficiency for biomass or food generation, with the rest of the energy getting lost to things like metabolic processes, inefficiencies, or environmental responses. For example, under certain conditions, up to 70% of the absorbed light gets converted to heat and lost. This is that sunscreen process that I was telling you about before. And so, in many conditions, it's actually that conversion to heat that limits crop yields. So if we want to understand this energy allocation and these limits, we can go back to our crop field and we can zoom in. And what we find are some of the most sophisticated nanomachines on Earth. These are the photosynthetic supercomplexes that collectively capture light and then convert that light into chemical fuels. These chemical fuels are then used for biomass generation. Now, even in the first steps of this process, this energy allocation is already occurring. Specifically, this is where we see the allocation of energy into these environmental responses, converted to heat and lost. But the size of these slices of pie, they actually change with many factors. And there's been decades of work in plant biology, spectroscopy, plant science that have all led us to understand that, for example, with something like weather, this amount can decrease dramatically. But if what we want to do is understand what it is that's controlling this, so we can begin to think about manipulation, we really want to understand the specific proteins responsible. So, for example, in green algae and moss, these are good model systems for understanding energy allocation in green plants in general. The specific protein responsible is one that looks like this. Here's the structural model. It's called light harvesting complex stress-related, or LHCSR. This is a protein that actually takes that sunlight and converts it into heat. And the level of that conversion gets controlled by that protein by structural or conformational changes. So if what we need to understand is energy allocation in photosynthesis, what we want to know is what is the conformational switch in proteins like this one that activate the dissipation of energy as heat. And so if we want to understand this dissipation of energy as heat, a really important reporter is fluorescence. So fluorescence is something that's used to study photosynthesis on all sorts of length scales. This is a wide interdisciplinary community. So what happens is proteins like this one absorb light. The amount of light that comes in then can come back out as fluorescence. So the more light that comes out, the less energy is getting converted into heat, and vice versa. Now, based on this inverse relationship, this is how fluorescence is used to study photosynthesis. And like I said, on many different length scales, from the kind of individual proteins experiments that we do in my group, all the way up to the global scale, where fluorescence is measured in satellites from space. And that really allows us to connect these different scales using these different approaches to understand energy allocation. But say we want to understand these individual proteins and this conformational switch, that question I posed before. Well, one challenge in a typical biochemical experiment is we may take proteins like this one and measure around 10 to the 4th individual proteins at a time. Well, the challenge with that kind of experiment is that each one of those proteins may not be in the same conformation or may actually be undergoing a conformational change. And so experiments had traditionally averaged over this, making it impossible to identify these conformations and the different energy allocation. Now, it's because of that limitation that over the past decades, single molecule, or in this case, single protein spectroscopy has emerged as a powerful tool. This is one of the major tools that we use in my research group, where we can zoom in and measure the fluorescence of one protein, and then another, to collectively understand all of their fluorescent properties. So here's an example of some data from my lab where we look at the fluorescence distribution. And so what we see are two peaks, one of which wears more of light harvesting conformation, and the other is our dissipative conformation, where the energy is being converted to heat. So this one has less fluorescence coming out. And so this data was taken under conditions that mimic low light, but we can change how we're doing our experiment to understand how energy allocation in photosynthesis changes. And so if we do the same thing, mimicking high light, what we see is a change in our distribution and an increase in the level of energy going to heat by around 50%. So more energy gets lost. So collectively, this allowed us to identify these two peaks, and see how this switch gets turned. So we now have this understanding of these individual protein conformations, how they change, and how they're controlling energy allocation in photosynthesis. And so with this fundamental understanding of the protein level properties, we can now begin to think about, that gives us a blueprint for what's happening in organisms like this one. So we can think about reallocating this, redistributing the energy, potentially to increase biomass, and maybe even one day increase our crop yields. And with that, I'm going to turn it over to our next speaker, who will talk about continuing to think about how to change these organisms, Dave Desmarais from Civil and Environmental Engineering.
Thanks, Gabriella. So I'm a plant biologist, and so Gabriella has just given you a fantastic introduction to one of the most amazing and central things that plants do for the entire biosphere, which is to take solar energy, this abundant but transient source of energy, and to capture that energy through the process of photosynthesis, interacting with CO2, with water, to make sugars. Okay? And as probably even most elementary school students know, the other thing, of course, that comes from this is also very important for the biosphere, which is oxygen, biomolecular oxygen, which we can breathe and which allows respiration to be such an efficient process for harvesting and using energy. So when I like to teach my introductory students in biology that we're taking this transient source of energy and we're trapping that as the bonds in the sugar molecules, right? So now we have this physical thing that a plant or an animal can use to transport, to move around, to use as energy when they would like to, not necessarily when the sun is shining. But as you probably know, sugars play a second equally important role, which is those initially fixed carbon molecules form the backbone of everything else. And I literally mean everything else, right? Everything that the plant makes, everything that an animal might eat from that plant, everything that a fungus might use as it decomposes that plant, all of those organic biomolecules begin as carbon photosynthate. But there's also very important other molecules that are part of that fixation process. So you know we need to eat protein because protein contains things like nitrogen and iron. Our lipids, of course, are often very complex, large macromolecules. And not only the composition of those molecules depends upon other nutrients, but also the processes to generate those molecules. The very action of doing photosynthesis requires all sorts of interesting elements that are largely gained from the soil. So what is the ultimate source of those important components? Well, there's many, many elements that organisms need, plants and us. Nitrogen is often most thought about. There's also countless mineral molecules. Where do those come from? Well, ultimately, those are weathered off of rocks. Okay? There are abundant sources of many of these minerals in the lithosphere, and as rocks weather, those enter the soils. Those can be captured by the biosphere, captured by plants and microbes to build their biomass. Nitrogen is also incredibly abundant, but it's abundant in our atmosphere as nitrogen gas, N2 gas. And you may know that nitrogen gas is incredibly hard to break apart those two nitrogen molecules, but breaking apart those two nitrogen molecules is critical to turn that nitrogen into usable forms of nutrients like ammonia, nitrite, which plants can uptake, turn into proteins, thus providing us with a food source. Now, in natural communities, which I have grown up studying as an evolutionary biologist and ecologist, in natural communities, where plants and animals and microbes are found and where they thrive is really a function of what's in those environments, right? If there's rocks nearby that can weather out these important minerals, well, then the kinds of plants that can make use of those minerals or that do well when they're in excess or in deficit. And similarly for nitrogen, where there's enough nitrogen in the soil, there's plants that will thrive in those places. And if they don't thrive in those places, in those natural communities, well, then they can move someplace else or they might go extinct. So natural communities have evolved over hundreds of thousands and millions of years to have compositions of plants and animals and fungi that form the biosphere, right? Some plants die, they rot, they create nutrients for microbes to produce, sort of continue the cycle of life. But in agricultural systems, we don't have that same nice balance, unfortunately, right? It's not simply a matter of having the plants grow someplace else because we want to have those plants grow at very high yield in the places where we need them to be growing. And so that creates a real problem when any of these things become out of balance. And this has led to what has been called the nitrogen problem, right? So we know that nitrogen is a key limiting nutrient for most of our agricultural production. Most of our calories in the world come from cereals, either that we eat directly, corn and wheat, or that we feed to our animals, which we then eat, the animals, okay? And to maintain these very high yields from cereals, we need to have a lot of nitrogen fertilizer. As I told you, those things can become limiting. If you imagine a common agricultural field, you're pulling up all that biomass, you're shipping it off, and the nitrogen is going with it. So we're constantly adding more and more nitrogen fertilizer back to the soil. The predominant source of the nitrogen in the developed world is through industrial fixation of nitrogen through the Haber-Bosch process. Here's a few figures to sort of make the point to you that this is an incredibly resource-intensive process to capture that atmospheric nitrogen and turn it into ammonia that we can feed to our plants. So just huge proportions of the total global carbon budget come from the process of making industrial nitrogen. But even more depressing is that most of the nitrogen we put onto our soil doesn't end up in our plants and doesn't end up in us as food. It's running off, or it's being lost back to the atmosphere as climate-changing gases. So this is the nitrogen problem. Now, you're probably aware that there are plants out there that have, in a sense, figured this out, right? So in places where nitrogen in the soil is very limiting, microbes can fix that nitrogen, atmospheric nitrogen. And many plants have figured out how to coexist with those microbes and make use of that nitrogen. So there's this really nice exchange of carbon from the plant and nitrogen from the microbes. And of course, legumes like soybeans and our many other beans are the most famous and widespread examples of this in our food system. But cereals don't have this trick. They haven't figured out how to have that symbiosis, how to get these relationships with microbes to provide nitrogen. So this has really been sort of a moonshot genetic engineering project since at least the 1970s, and a number of proposals have been brought forth about how we might try to introduce this nitrogen capture process into our cereal crops. So at base, what this issue is how do we get the nitrogen out of the atmosphere into a microbe and into the plants we want those microbes to be associating with? And in turn, what is the plant then giving back to those microbes in exchange for this resource, this valuable, energetically intensive resource capture? Okay. So this is sort of the central question. And this is a challenge that we've been tackling as part of the MIT Climate Grand Challenge program, led by Chris Voigt, a number of faculty from across the institute, where we're trying to address this nutrient use in agriculture problem in a very holistic way. And what I want to share with you today is a small piece of that project, which is trying to optimize this particular little interaction. How do we get those microbes to capture the nitrogen, give it to the plants that we want them to give it to, under the conditions that we need them to do so, and then have the microbes, in a sense, cultivated by the plants? So Professor Chris Voigt here in MIT's bioengineering department is really focusing on this first question. How do we genetically engineer bacteria to fix the nitrogen when and where we need them to do it? My group is really interested in the plant side of this. My group is comprised of plant physiologists, plant geneticists, and we think very deeply about how plant performance is driven and affected by environmental conditions and environmental change. And one thing we've been focusing on the last few years is trying to understand under what conditions do plants provide these cheap carbohydrates, organic acids, and sugars to the microbes in the soil. Is there genetic variation in this process? How does the environment drive this? And critically, could we understand at a mechanistic level how to manipulate that process? Okay. These are some really key questions we're interested in. What molecules are there being produced? Does the environment drive this process? And critically, can we manipulate the production of what we call these exudates? Now, to do this requires a tremendous amount of technological know-how. So we're looking at roots, so they're below ground. We can't just walk up to the plant and measure them without disrupting itself. So we're collaborating with a Professor Sishan Yu here in MIT's Research Lab for Electronics and EECS, who's developed non-label, non-invasive imaging technologies for living tissue, where we can look at the roots in situ as they're growing and look at their metabolism, look at their chemistry, to see how they're growing at the cellular level. And my PhD student, Chloe Heitmeyer, is developed a very simple system to grow these plants under controlled conditions where we can actually then image them in situ and critically sample those exudates from living plants. So as I introduced at the beginning, I'm a plant biologist, right? And when I talk to my colleagues at the universities, they say, well, what on earth are you doing in this environmental engineering department at MIT? And this is what I'm doing here, right? The opportunity to collaborate with electrical engineers and to recruit the best PhD students in the world to come and work in the lab is why we're here. It's this interdisciplinary fearlessness that I think so many of us here at MIT bring to the table. And I hope that we can use this integrative approach to tackle this very simple but very complex mechanistic problem in a way that we can hopefully advance food security and ultimately human health. So with that, I'd like to introduce Professor Janelle Knox-Hayes, who's a professor of economic geography and the planning head for MIT Architecture and Planning.
All right. Good afternoon, everyone. Thank you so much for joining this panel today. I'm going to present some research that's looking at the interface, kind of taking a scale out at the city level, the interface of climate impacts like extreme heat and health dimensions. And so This is based out of work that was completed a couple summers ago in the height of the COVID pandemic with Cynthia Rosenzweig at Columbia University and Juan Camilo Osorio at Pratt University. And we were really interested in understanding what was happening with the pandemic and how city officials could respond to interventions such as extreme heat in the summer of 2020 and 2021 when the pandemic cases were at their highest, but people were not able to be brought together into cooling centers. A lot of public infrastructure wasn't operational, and so we wanted to look at those dynamics and see how extreme heat is really a factor of a range of different, not just natural conditions, but built environmental, socio-environmental conditions. So our questions for the study were, what are the places of highest risk and heat for COVID? And we focused on New York City in part because Cynthia and Juan Camilo are based in New York City. We had really good access through their network works to a range of data, including COVID health data, and then also this satellite imagery. But New York City was also the sort of the ground zero of the pandemic for the United States. And what we quickly discovered is that the pandemic unfolded in waves, and we could track those waves. They were very distinct within New York City. So we basically mapped 3 waves of the pandemic, and that allowed us to look at the relationship between the environmental, the built social infrastructure, and the socio-demographic characteristics. And then we also wanted to understand the neighborhoods that were most impacted by COVID-19. We were able to show that there's a direct correspondence with compound risks of extreme heat. And though it's not yet published in this work, we also looked at factors like flood risk and air quality risk and show that those risks, they're compound. If you're vulnerable to one of these risks, you tend to be vulnerable to all of them. And so in some, we're looking at a range of economic inequality, health, built environmental factors, and natural environmental factors, including heat, extreme heat, air quality, and flooding, and creating a compound risk index for the city that they can use to make different planning decisions. So there's a lot of data here. I know I don't have a lot of time to try to walk through this, but I'm going to try to walk through some of the key findings from each of the waves of the pandemic, and then look at our index and how that can shape different public understandings of how to respond to things like the COVID pandemic. So the unique challenge, again, the cooling strategies, such as cooling centers, did not meet the physical distancing requirements of COVID. Vulnerable communities had less access to green space. The other scientists on the panel have been talking about how significant different factors of plants are for generating food, for remediating heat, for generating energy, and we absolutely saw that in terms of vulnerabilities to COVID-19. We know a lot about the impact of crowding in the spread of respiratory illnesses like COVID-19, and that there's a general relationship between density, urban density, and spread. What was really interesting about our data, though, is we were able to parse urban density and show that the most dense areas of New York City actually weren't the most vulnerable. There are different kinds of densities, and so I'll walk through kind of what those results look like. And then we looked at the social determinants of health, including racial dynamics. We created a social vulnerability factor index that included things like language isolation, access to resources, education level, income level, and compared that as well with COVID cases. So just to sort of take a quick look at New York City, we're mapping the 5 boroughs. And I don't know if I have a pointer on here, maybe I do, yeah, where we can see kind of Bronx, Queens, Brooklyn, Staten Island, and then we combined to understand vulnerability the first 2 waves, because there's different factors that occurred across all 3 waves. In the first wave, initially, people were being told to stay indoors, not to mask. Later, that was reversed, and we realized actually being outside is much better if the weather is good and you have access to green space. Get out into fresh air, don't be crowded into indoor spaces, and masking became essential. In the second wave of the pandemic, there were lockdowns across the cities, and you can kind of see here the timing where we were able to map each of these. Oh, sorry, my pointer isn't quite. First wave between January and July of 2020 was a summer wave. Second wave between August and July of 2021 included the peak in a winter wave. And then between the second and third waves, the biggest intervention is this is when vaccinations became available. So you had a much more extreme variant of the virus in the third wave, but we'll look at sort of how vulnerability played out through vaccinations. All right, so the study design, really, this was taking inspiration from an epidemiological study, Madrigano, who created a heat index for New York City and creating a spatial variation of that. So our data are all associated. We had more than 30 variables. We associated them spatially rather than trying to think about cause and effect of the pandemic. We were looking at spatial correspondence. We created this compound risk index, and then we evaluated the interventions. And the way we did this was by combining the variables into different factor groups. So we looked at vegetation density to understand this relationship of green space. We looked at impervious surface because, again, it's not a matter of total density, and you really, in New York City, see that with the impact of Manhattan. Manhattan is incredibly dense, but it has, with Central Park in the middle, really good access to green space, and then other socio-demographic conditions that made it less vulnerable, at least in the first and second waves of the pandemic. That helped us create this idea of a mixed density variable, which we differentiated from rental overcrowding. So it's not just a matter of density. The mixed density in some of the places in Manhattan, it allows for this concept of a 10-minute city. People who were living in those conditions had access to childcare, to food resources, to quality green space. They might be within 10 minutes of their workplace, and that had a severe impact or a high impact in reducing the severity of COVID cases. And then also the distance, this was kind of critical, especially for those that didn't have access to air conditioning, to public cooling. And then finally, the social vulnerability, we combined different factors to look at social vulnerability. So, you know, some of the map on the left here shows the top 15 most vulnerable neighborhoods. And a lot of these were widely reported in the news. Kensington Heights on the north, and then, sorry, I'm not sure why this isn't quite working. Corona was one of the most impacted neighborhoods in Queens, and then Sunset Park in Brooklyn were some of the areas that were getting a lot of attention. And one of the first things that you notice is the distance of these neighborhoods from quality green space. So we used a number of different factors to map and measure green space and look at where are communities and what kind of access do they have. And then this is, again, quite different from impervious surface. It's one of the core vulnerabilities. You can see how physically dense Manhattan is, the relative impervious surface, but that doesn't necessarily correspond with quality green space because you have good access to green space there. I'm going to skip through the regressions. I think there's interesting results in the paper if you're curious to dig through that, and just sort of quickly highlight some of the key findings. In the first wave, temperature and COVID-19 cases concentrated in the same neighborhoods. So green space is not only for mediating heat, it's also for mitigating the impact of the pandemic. It was highly significant, especially in this summer wave where people needed access to quality outdoor space. Social vulnerability was highly significant across all three of the waves. And again, for those that are curious, you really can see this impact here. There were really interesting differences across the waves for, access to cooling infrastructure, as you can imagine, was significant. Lack of air conditioning was significant in predicting vulnerability. And then, of course, distance to open space and social vulnerability. In the switch between the first and second waves, we identified a really fascinating transition. It was known that Black and African American populations were particularly vulnerable in the first wave. Hispanic populations were also particularly vulnerable, but for whatever reason didn't get the same level of media coverage. And we were able to demonstrate with our data a shift that occurred between waves 1 and 2, where that vulnerability was largely addressed for Black and African populations in the second wave, and it was not addressed for whatever reason for Hispanic populations. We looked into the employment records, and there's actually, for both Black and African American and Hispanic populations, a higher majority that are working in essential services. For the Black population, that was primarily in transit. For the Hispanic population, that was primarily in cleaning services. So there may be some factor there that's not fully explained in our data, but should be looked into as to why one population was more vulnerable. And then the rental overcrowding, we saw a huge impact there with the difference between rental overcrowding and mixed density. And so this map, I just want to quickly illustrate the differences here. This is the first wave mapped over Black and African populations. You can see how strong the direct correspondence between COVID cases and those percentages. And then this map is the blue hashes are showing the second wave of the pandemic and Hispanic populations. And again, you kind of see, especially in Brooklyn and Queens, shift away from predominant Black and African populations, but the direct impact on the Hispanic populations. All right, and I'm almost out of time. I know there's so much data in here to cover. Rental overcrowding, again, huge overlap and prediction with COVID vulnerability, which was, again, very different for mixed density in the first and second waves. What's interesting is in the third wave, we really see the impact of the vaccination rate. This had a profound shift in moving the vulnerability. So you kind of see the areas in Queens that were deeply impacted in the first and second wave are less impacted in the third wave. Manhattan finally had a bigger impact in the third wave, and this may be as a consequence of people's choices around vaccination and where that was directed. And so just to conclude, I know I'm out of time, but the way that we utilize this is we combined all of the data and created this risk index for the city so that they could see where are the areas that are most vulnerable. This part, the oceanfront areas around like Long Island are very well understood for flood risk especially. But this back bay of New York was not well understood how vulnerable these populations are here. And then we went through and looked at the existing interventions for heat, open spaces, pools, open and cool streets, and mapped the correspondence of areas that are high risk and have low access to services, the areas that are high risk but have good access to services, and then the areas that are low risk and low distance to cooling, and presented these findings to the city so that they could look at mapping their interventions and understanding where were existing vulnerabilities and where should they better be targeted. All right, so I'll leave it at that. Thank you very much to Lisa. And I'm happy to pass it now to our next speaker from Biology Institute Professor Penny Chisholm. Thank you.
Thank you. I have a couple of props here, which I'll explain in a minute. Well, it's sort of obvious, but so I am going to focus on the big picture with this talk, and we're going to go back to photosynthesis. What I'd like to do is, first of all, show you my favorite slide on life on Earth, and I'm going to describe my passion for phytoplankton over the last 40 years, and I'm going to use that as an example of how curiosity-driven research can yield really interesting understanding of life on Earth. And this is just going to be one organism I'm talking about, and there are millions of others that we should be studying. So that's my pitch, and then finally I'll make a big pitch for that for MIT. So first of all, life on Earth, this is my abridged version. We can't forget that life all depends on this process of photosynthesis. Well, there's some microbes that can make biomass from deep sea vents, but if you track that, they need oxygen, which comes from the surface. So all of life depends on this photosynthesis, taking solar energy, CO2, and turning it into food and fiber. And then all of the organisms on Earth that can't do that, take that, we eat and we respire and use that chemical energy to live. And I felt so passionate about this, I actually collaborated with a friend of mine. We wrote a children's book called Living Sunlight, and it starts out, it's narrated by the sun, and it starts out with a child, whoops, with its hand. It said, the sun says, put your hand on your heart and feel your heart pumping, feel how warm you are. That is my light inside of you. And I showed this to a friend of mine when we were drafting it, and she said, oh, that's a beautiful metaphor. And I said, it's not a metaphor. I said, that is sunlight, energy in there. And I just think it's so important to think about that, because half of the MIT plant, well, I digress, but half of the MIT plant scientists are in this room, and we need more of them. That's one of my messages. Okay, so Now, on today's Earth, these two reactions are fairly balanced, roughly. But if you integrate over millions and millions of years, there's more productivity than there is respiration, and we have this fossil fuel, which is basically ancient plants. That's buried sunlight, another book over there. And what we are doing in this 150 years is taking that and burning it, same process there, respiration, and that's the CO2 that's increasing dramatically in the atmosphere. Hence climate change, hence MIT's huge initiative on climate. So all of these initiatives are in this one slide. But today I'm going to focus on that neglected little green thing up there, which is the microscopic plants, the phytoplankton, that do actually roughly half of the photosynthesis on Earth if you round up, maybe a little bit. And oh, you can't see this very well, but it's from another children's book on the phytoplankton. But the there they are, these little microscopic green plants in the upper 200 meters of the ocean. This is roughly to scale. And they feed all of the life in the ocean ecosystem, even the life in the deeper waters. And that is through the settling of the biomass that is not eaten in the surface. We call this marine snow. And so it's dead stuff and mucus and everything that's colonized by bacteria. And as it settles, of course, there's no photosynthesis down there, so there's no consumption of carbon dioxide, but there's respiration, so the bacteria are respiring carbon dioxide. And what that does is it sets up this concentration gradient. It's pumping CO2 into the deep ocean. This is called the biological pump. So phytoplankton serve this function in the oceans of constantly keeping the CO2 concentrated in the deep ocean. And just as a thought experiment, if they didn't exist and you mixed all the ocean, so all that CO2 equilibrated with the atmosphere, there'd be 2 times as much CO2 in the atmosphere. We'd be really cooked. Okay, so when I came to MIT, I was already studying phytoplankton, and most of the different species, here's some pictures, are roughly were between 10 and 100 microns in diameter. In 1979, a colleague, John Waterbury, in Woods Hole discovered these tiny little 1-micron phytoplanktons called Synechococcus. They're shown here fluorescing orange because of the pigments they have. And they were discovered because a fluorescence microscope suddenly became available, and you could see these tiny little things. And I was lucky enough to have a team of people, to work with a team of people. We took a flow cytometer, which is a laser-based instrument on a ship, and Rob Olson, brilliant young scientist, was able to see even smaller than Synechococcus. You can't really see it. There's 2 little red dots there. That's also chlorophyll fluorescing. That circle, this is this little organism called Prochlorococcus. It's the smallest and most abundant photosynthetic cell on the planet, it turns out. It's less than a micron in diameter. It's the size of the wavelengths of light that it absorbs. And I've been studying it ever since. It's been my muse. And we now know that there are 3 billion Prochlorococcus in the global oceans. They're everywhere in the warm mid-latitude oceans. Their annual photosynthesis is equal to that of all crops on land. And over the years, we've been able to isolate them into culture. And after the human genome was finished being sequenced, we were able to start. They were one of the first microbial genome sequenced because they're so small. You could do it in half a year. Now you can do it in a half day. It's very exciting. So we're knee-deep in genomes now. So genomes, transcriptomes, we use all of the technologies that are developed for biomedical research. We apply to Prochlorococcus. And now we have a database. We can go, these are transects in the world oceans where we have a database of Prochlorococcus genomes from all over the global oceans. And so over the years, I just let Prochlorococcus point the way in terms of what we should study. And this is a beautiful model organism to use for what I call cross-scale biology, which is really ecology, but that word is loaded, so we call it cross-scale systems biology. And so my goal was just to use this organism to study it not only at the molecular level, but all the way to the global biosphere level, the oceans, which you can do with this simple little cell. And some of the things, these are just some of the stories that we've learned along the way. The first thing we learned is that although this cell has about 2,000 genes, every time you sequenced a new one, you'd find 100 new genes that you didn't see before. And the projected pangenome of the collective of all Prochlorococcus in the oceans is about 80,000 genes. That's 4 times the size of the human genome. So this tiny, simple little cell, it's really a superorganism. And we can debate whether it's a single species, but by many measures, it's a single species. So it's incredibly diverse, and that's how it maintains these populations throughout the oceans. Also, one of my postdocs recently discovered a novel gene transfer agent, which he named tachyposons, after a goddess of good fortune or something. Anyway, these serve to move genes around within the superorganism to continuously create the diversity in the system. They also make these novel metabolites, secondary metabolites. They make hundreds of these molecules that in most model microbes that are studied make 1 or 2, and we don't know what their function is at all. That's somebody else has got to figure that out, but it's really exciting because the cells are trying to tell us something. There are thousands of these molecules in the ocean. So we see them in the cultures. You go in the ocean, look at the sequences, and you can see that there are thousands of variants. Also, we've studied vesicles. They pop out little lipid-bound vesicles that contain DNA and RNA. So then you go to the oceans and you say, oh, are there vesicles in the oceans? And yes, there are. There are thousands of different species are making these vesicles in the oceans and putting them out there. It's like dissolved information going around in the oceans. And also studying interactions between Prochlorococcus and other organisms. A new evolutionary theory was developed, not in my lab, but in a colleague's lab, called the Black Queen hypothesis, which is really interesting. So some people say, well, that's all very interesting. It's basic research. But how can we use them? And I get this question all the time. And I say, well, they're out there doing their job, you know, keeping the biological pump and all of that. But I look to the San County Almanac for the answer from Aldo Leopold, and hopefully not insulting anybody in the audience who wondered how to use them. But I'm sure that in time, they will be used. They are the smallest amount of information that can do this amazing photosynthesis reaction. Okay, so what do we need more of at MIT? I think we need more of this curiosity-driven research on life on our planet. And I'm all for human health, and that's what this initiative is about. But also, I think we need to broaden our scope of life sciences to embrace, most importantly, plants. I think we need more of that at MIT. So life is a complex adaptive system, and here's my laser. This is where we've focused a lot of energy here in biology at MIT for good reason and with amazing results. I think that focus is incredibly important. But what I like to remind us is that this is how things work. But this, at this level, the ecosystem level, population level, is why they work that way, because that's where evolution is operating. And there's feedback mechanisms from this level of organization feeds back on this level of organization and shapes it through natural selection. So we need to look at both the how and the why of the system. And this movement is underway. I'm very excited to say. I say let's not miss out. Molecular biologists, these are papers that just came out recently. Let's reconnect with nature, integration across scales. This one is actually from a colleague at MIT, Mick Follows, a metabolism shared across scales. So there is a movement to go in this direction. So with that, I thank Prochlorococcus and all the people that contributed to this. But most importantly, 2 foundations made all of this work possible. The Moore Foundation and the Simons Foundation. I never would have been able to do this without that support. It's extraordinarily important. And finally, you probably can't read this, but I was playing around with ChatGPT and said, what would MIT and Kendall Square look like in 25 years if my fantasy world existed? And that's what we hope will happen. And it came up with plants all by itself, so it understands. Oh, I'm supposed to sit down.
Okay. Fantastic. Let's see. So I really enjoyed these talks. I hope you did too. I think it really captures how life science, broadly defined, is going to play huge roles in many aspects of human health. And these are leaders in that space. So this is your chance to ask them questions. And I heard there's a prize.
First question gets a free book.
You do have to come up to the mics to ask your questions. So if you can come up to the mics and ask your questions, that way the interpreter can hear and interpret. Thank you.
Okay, I have actually 3 questions for 3 different people, and they're quick. One is, given that the nitrogen fixation requires the combination of a plant and a microbe, is anybody talking about simultaneously engineering both of them, both kinds at the same time to communicate? Second question, the density that you cite in New York, do you consider per area or per volume? I mean, New York is really built up, so that might make a difference. And the final question is, in deep sea vents, is it really dependent on the sun?
I guess we can tackle them in order.
Yeah, sure, we can take them in order. Dave and then Janelle.
So yes, so the way we're envisioning this is sort of a co-optimized system. We're not using sort of any sort of directed evolution process where we're the process is not so much co-optimized as the goal to have a co-optimized system where we understand what the plants want, what the microbes want, and how they're talking to each other. So yes, the idea is to have them synergistic and sort of designed in tandem to be together.
Yeah, and on the density question, it's a terrific density, or a terrific question. We were looking at density in terms of the total population density, and then for mixed density, it was including building type and building use. So mixed density included both residential and commercial. And when you map out what that variable where it is, again, you see kind of the areas like Manhattan have a lot of mixed density. Rental overcrowding was also taking total population density and combining it with housing type. So not type of family, is it a mixed family, multiple family unit complex, and then number of residents in each unit. So rental overcrowding showed areas where you have rental use, but then more density in each rental unit. So kind of, I think that is both combining the area and the volume for that question.
The deep sea vents, the bacteria down there are oxidizing hydrogen sulfide, and so they need oxygen to do that, to fix CO2, right, Dave? Yeah, so it's coming from, it's coming from the atmosphere up above, which is coming from the plants and finding its way down to the deep ocean, and that, so in effect, they need oxygen from plants.
Thank you. Hi, my name is Amy Nernberger. I'm from Data and Specialized Services and Research Data Management. Thank you, each of you, for such delightful presentations showcasing the variety of work that is done around the biosphere and plants. Given that necessary interdisciplinarity, the focus on collaboration in this initiative, can you each speak briefly about what you would like to see from yourselves, from your colleagues, from others on the stage to increase the effective sharing and reuse of the data and other associated products that your research creates so that you can more fruitfully grow from each other? Thank you.
Well, I think, one of the themes of this session is across scales. So I think the key to that understanding, and Penny did a great job of wrapping it up with that, is understanding how to get the output of one scale to merge the input of the other and vice versa. And I think sometimes fields are very specific in the type of data that they produce and how they look at it. But I think finding metrics, like in some ways, actually, I think photosynthesis is a great example for this because of fluorescence, like we both talked about, right? That actually allows a kind of cross-scale, because you can look at the process itself in a way that you can't with other things. You need a exogenous reporter. And so I think while we have that in what we measure, we maybe don't save it in the same way or report it. And I think having catalyst type funding that allows you to kind of build across fields allows that kind of thing to happen.
Yeah, so I mean, your question kind of keeps me up at night, to be honest. So I'm a, we heard a lot about omics this morning. Penny mentioned different omics. I do a lot of genomics. And we've had like 20 some years now to think about what to do with genomic data, right? In a sense, it's kind of easy in that there's only 4 letters that make up, well, 5 if you want to count, uracil for RNA. But it's something that's pretty easily stored and manipulated, and we more or less understand what it is. And we've now gotten to the point where we can collect all the other omic data much more easily than we can analyze it or even handle it. So I am very interested and very concerned with how we would sort of standardize metabolomic, proteomic, phenotypic data, the kind of stuff that Gabrielle's lab generates. Like, these are, optical things. Like, how do you quantify and store that and compare it? So thinking about ways to integrate big data and be clever about how we generate the big data, I'm very bullish on, and I think we need to spend a lot more time on doing it. I'm optimistic that people at MIT can sort this out, but it's something that we're very focused on.
Yeah, so another area of my work I wanted to talk about today, but didn't really have time, is indigenous community planning. And I've been building a group in urban studies and planning that's really focused on trying to weave traditional ecological knowledge into conventional sciences. So for example, the nitrogen cycle, it's a well-known in indigenous practice of growing 3 different species together, the corn, beans, and rice, to help fix the nitrogen. There's a lot of knowledge about plant, urban, and natural systems that comes from indigenous communities. So the green space we showed in the COVID study, how significant that is. It has a cooling factor, but it isn't just the physical cooling factor. There's a lot of well-demonstrated health literature about how being around high-quality green space or high-quality density of trees helps human health. And one of the things one of the students in my lab has started studying is seeds, the way in which they have actually acoustic resonance. So she played sounds to seeds and then has demonstrated that the seeds actually respond. There is sound that comes back, and she's done some really amazing architecture and art displays mapping and showing what that sound looks like. But we're partnering now with Professor Nicholas Macrease, who's in aeronautic and acoustic engineering, to look at that sound resonance across a broader spectrum of trees. And it could revolutionize the way we think about urban planning in relation to plants. So there's a whole, not just within the seeds, but mature plants, a whole sound biology that exists around them. And we're in planning often thinking just about sight visually, what are we seeing, or even the light impact is somehow a sight impact, but acoustically what is happening both in our natural and our urban environments is critically important. And so this kind of combination of indigenous knowledge, what is known about the species, what they're speaking, what they're saying, the kind of the urban planning, and then the life sciences of understanding the physics and the engineering of that could be really critical in unpacking and creating new areas and ways in which we think about planning and development and health. And so I think this initiative really captures that spirit, and that's critically what we need more, is to put people from different disciplines together, from different ways of knowing together, and combine that knowledge to better effect.
To be efficient, I'll just second what Dave said, because we have just this extraordinary database of Prochlorococcus sequences from all over the global oceans, just screaming with studies, with things to tell us. And it's just a huge challenge getting it all organized so that others can use it.
So we have time for one more question today, which is fantastic. You guys didn't have to listen to any of my questions, so well done as an audience. Go for it.
So my question is about the COVID-19 research. I'm curious, I think it's Lydia Borriba's lab that showed real correspondences between humidity in the immediate environment and COVID transmission, and that, you know, in tropical locations, it's when the humidity goes above a certain point that COVID transmission increases, and in climates like ours, it's when we start to spend a lot of time in dry heat in the winter that COVID transmission increases. I was really struck by your mapping of flood zones and heat zones with COVID, and I'm curious if that is, if there's any collaboration there, if that's another factor, and if that sort of humidity might be another interventional lever that we could use in public health.
Absolutely, that's a terrific point, and thank you for highlighting that research within MIT as well. It's something we haven't published the flood risk paper yet, the flood risk version of this, but something that was really significant in type of building was basement use, like where are people living in basements? It's a huge vulnerability. I mean, you can understand for flood risk, obviously we've seen that in New York City before with some of the major storms, but then also with COVID risk. And so humidity might be one of the factors where, again, kind of where are people living in crowded spaces, rental spaces with suboptimal urban conditions, and then how is that increasing the compound risk or the compound vulnerability? A great point, thank you.
Thank you.
Thank you guys very much. Thank you for being here. You have 20 minutes to make your way back over. So thank you guys.